Hello and welcome to this video on the control of the cell cycle. This has to do with the regulators that keep the cell from going crazy. And so a cell that's gone crazy, like a cancer cell, where, a lot of times, very, very frequently, what causes or what doesn't keep this from happening are some of the mechanisms that we're going to talk about in this video. We are following Clark, Choi, and Douglas's Biology 2E, and this is the third video on cell reproduction. So we've done a video on mitosis, Gesundheit, and also on kind of the basic structures of chromosomes and things like that. So this video is on the control, the regulation of the reproduction. So the section starts off by talking a little bit about the rate at which things reproduce. So if you're a human embryo, you're going to be reproducing, every few hours, an embryo is going to duplicate, get twice as big, I suppose. And so in a few hours, a human embryo will reproduce. Now, epithelial cells, like in the inside of my cheek, they may reproduce every 2 to 5 days. So that's a little bit slower. And then there are, of course, some cells, like some nerve cells, that will never reproduce, like your brain and so forth, which is why I'm not as smart as I used to be. But anyway, so the rate of division varies from animal location, what I mean is like the kind of organism it is, and also the type of cell that it is. Now, the stages of reproduction also vary. So if let's say that a cell is going to reproduce in a 24-hour period. So what percentage of each stage would it be at? So if a cell were to reproduce in 24 hours, then the G1 interphase part, remember there are 3 parts to interphase. The G1 part of interphase would be 9 hours of that 24. So that's what? It's less than a third, but it's still a hefty chunk. Remember, the G1 phase is we're getting the materials together. We're not ready, but we're getting them together. There'll be a kind of a checkpoint at the end of G1 to decide, are we going to go forward with this, guys? But G1 would be about 9 hours. So the S phase, that's where the DNA is replicating. The replication phase, that's going to take about 10 of those hours. So that's what, 5/12. Little less than half of it is spent in the replication phase. And then G2, about 4.5 hours. That's a shorter period. And then the actual mitosis phase, the actual part, would only be like a half an hour of all that. So very little of the cell's time is taken in the actual pull part of it. That most of it is in this kind of in-between part where it's getting ready to rumble, okay, in the interphase. So that's kind of a sense of the relative rate of the stages. Okay, here's a nice little diagram. I like this diagram. So if we think of this point right here as where we've just finished reproducing, high fives, everyone, so G1, get materials ready, is going to take up quite a bit. And then there's a checkpoint. There are 3 checkpoints where we say, are we going to go forward with this? We just want to just check everything before we go any further. So one of them is at the end of G1, and then this would be the DNA replication phase, and then G2, getting ready to divide. There's a checkpoint at the end of G2. So at the end of G1, at the end of G2, there's a checkpoint. And then at the end of, metaphase, sorry, went blank there. In metaphase, right before we start so remember, anaphase is the tug of war, pull. So anaphase is where we pull the chromosomes apart. Right before that, in metaphase, when we're hooking up the spindles, right? Everybody's lining up. Everybody's lining up in metaphase. Before we start the pulling, Everybody lines up in metaphase. Yeah, the spindle part is pro-metaphase, right? I hope I'm getting that right. Let me double check real quick because I don't want to mislead you. Yes, that is correct. We hook up the spindles in pro-metaphase, and then everybody lines up in metaphase, and then anaphase is, so, okay. So there's a right before, at the beginning of metaphase, there's a checkpoint to make sure all the spindles are hooked up. Okay, so what does each phase check? I've already kind of told you. So at the end of G1, the question is all the DNA intact? So is all the DNA good to go? Because before we replicate the DNA, we want to make sure that it's all good stuff. We don't want to replicate bad DNA, right? And so there's a checkpoint to make sure, is all the DNA good here? Because we don't want to reproduce until all the DNA is good. So the G1 is the DNA intact. And then at the end of G2, the question is, do we have duplication? Has everything been copied here? Is there anybody that hasn't been copied? Everybody copied. Okay, so G1 is, do we have good stuff to copy? At the end of G2 is, has everything copied right? And then, like I said, in metaphase, is everything on a spindle? Everybody hooked up for the pull, everything ready? Everybody ready to line up? Metaphase is where they line up. Okay, so this is what the checkpoints check. Checkpoint Charlie. In more detail, again, I've said it, I'll say it again. Review is good for the soul, right? Say it 3 times and you've got it forever. Maybe not, but I like to say that. Are we going to do this? This is the G1 checkpoint. Are we, is the DNA intact? This is the restriction point in, say, yeast growth. Are we going to do it or are we not going to do it? Check for damage. Anything? DNA look okay? Everybody look okay? Great. Okay. G1 checkpoint. Is the DNA good to go? G2 checkpoint again. Are all the chromosomes in here replicated? Everyone in the car. Okay. Is everybody here? Did we leave anybody? Everybody got one? Okay. Is your brother here? Where's your brother? And then the M checkpoint is the spindle checkpoint. Everything hooked up. It's kind of like on the roller coaster ride. The people make sure all the seat belts are before the roller coaster starts. We got to make sure everybody's hooked up and seat belted. So are all the kinetochores? Kinetochores are the kind of the proteins where the spindles stick. Everybody hooked up for mitosis? This is karyokinesis. Just to remind you of things, that the mitosis is karyokinesis, the movement of the karaoke. Okay, so metaphase is where this takes place. Okay, 3 checkpoints: end of G1, end of G2, M, mitosis. Metaphase. Is the DNA intact? Is it all replicated? Everybody hooked up? Okay. So there are kind of regulator molecules. And the regulator molecules, there are 2 kinds of regulator molecules, okay? There are positive regulators and there are negative regulators. So this is about the positive regulators. The positive regulators push the process. And so these proteins called cyclin proteins, you can see there, it looks like there are about 4 basic cyclin proteins. There's cyclin D that seems to be involved with kind of the whole process. Then you've got cyclins that become involved with specific phases of the process. So cyclin E is the protein, and by the way, I think of cyclin, I'm moving. These are trying to keep it moving. So they're cyclin. They're revin. They're cyclin. And so what a convenient name. And so you can see that when we are trying to decide, is all the DNA good? There's a spike in the cyclin protein E that helps make sure that all of the DNA is intact. And then cyclin A seems to be involved when we're coming to the G2 phase to make sure that everything is properly replicated. And then there's cyclin B. I don't know where cyclin C is. There are multiple cyclins. This is a multiple cycler race. But that gives you a sense of these cyclins that are involved. They're positive regulators that say, let's go, guys. Now, the cyclin proteins attach to things called CDKs, cyclin-dependent kinases. So ACE is the enzyme, right? They're my kin. And so these two go together. The proteins bond, and here's a picture of it. Let me move my face out of the way. I hope that works. So you can see that the cyclins attach to the cyclin-dependent kinase enzymes, and this connection helps promote the progress of replication. What happens is a phosphorus is like the money. So if this is a machine, first of all, we hook them together, and then you put coins in the machine to get it to go. And the phosphate is what is the coin that makes it happen. And then, so this works, the process works by phosphorylation. Phosphorylation, again, is the donation of phosphates. And so these two attach, a protein donates a protein, put a coin in the machine, and then the protein that's going to help it happen comes up and fills up with gas. The phosphate is given to the target protein, which I've got gas now. So now this other protein can go and advance the cell cycle, can keep things going. So these positive regulators keep the process going by hooking up. They hook up and they take a phosphate, and then they give the phosphate to a protein, and the protein has enough energy. Let's go. Okay, so the process works by adding phosphate. So a phosphate activates by changing the shape of the protein. That's how it activates the thing. So there are also inhibitors that keep this from happening. So that relates to negative regulators. So we have positive regulators that get it going, and then we have negative regulators that say, whoa, we're not ready to do this yet. Okay, positive regulators get it going, negative regulators keep it from happening. And you need to have both functioning, right? If you don't have the positive regulators, it's not going to move forward. Or if you have the negative regulators, it's not going to go forward. And so they both work together. So here's some negative regulators. The negative regulators stop the cell cycle. And some of them just have, they don't really even have names, some of them. So there's one called P53, which means protein 53. And the 53 means that it weighs 53 kilodaltons. Basically, there are 53 protons in this thing. A Dalton, I didn't know this. A Dalton is a 1 AMU of mass. So an AMU, you'll remember, is an atomic mass unit. That's 1 proton. So a kilo AMU is 1,000 protons. And so if something is 53 kilodaltons, then it's 53,000 protons. That's a big molecule, it seems to me. But the way they discovered P53, if I understand correctly, is cancer cells have mutated versions of this. So one of the things that is a release the cancer. The thing that, well, that's a bad. Okay, I guess a kraken, release the kraken. Anyway, so one of the things that allows cancer to happen is when P53 protons are mutated to where they're not effective to, whoa, hold, stop reproducing. And so the P53, this particular protein is defective in cancer cells, which is why the cancer cells can blah, blah, blah. So again, P53 means protein 53, and 53 stands for the number of daltons or protons, as you would, or includes neutrons too, right? Protons and neutrons weigh about the same. Basically the nucleus, atomic mass units, 53,000 protons and neutrons in this particular protein. Anyway, more on that in a moment. More on it now. P53 acts when the DNA is damaged in G1. So that whole, at the end of G1, is the DNA good? Are we ready to replicate the DNA yet? The P53 is what stops it from moving forward at the end of G1. It halts the cycle and says, hey, wait, okay, we've got to repair some stuff here. We got some DNA damaged, bring in the repair enzymes. And so the P53, and I'm not sure how to help you remember P53, except that I'm 53 years old. Papa 53, I stop cancer. Yeah, okay, never mind. So it halts the cycle and recruits repair enzymes. P53 at the end of the G1 checkpoint. Okay, and of course, if it's irreparable, it causes cell suicide. Don't think of me on this one. But the name for cell suicide is apoptosis. Pop goes the cell. P53 may trigger apoptosis, Pop-Tart, apoptosis, apoptosis. Anyway, if the cell is not, I'm sorry, we can't do anything here. P53, blows up the cell. Okay, so P53, our first negative regulator. Okay, cyclins are positive regulators, right? Cyclin, moving forward, positive regulator. P53, Papa 53, stop. You're not ready to reproduce yet. You're not married. Okay, so rising P53, Papa 53, causes the rise of P21, which is Tom next year. But anyway, so rising P53 causes the rise of P21. P21 enforces the halt of reproduction. What it does is it binds to the cyclin CD, it disables the positive regulators. It stops the positive regulators. You guys are too positive. You need to stop down, I don't know, 21. I can't come up with a really good metaphor for P21 doing that. But anyway, P21 is triggered when there's a lot of P53 in the air. And think of Tom as an MP, right? He's going to stop this cyclin. Anyway, so P53, when it rises, causes the rise of P21, which again, 21 kilodaltons. That means 21 protons and neutrons in that molecule. It's a smaller protein. Okay, and the third, so we've got P53, Papa 53, and we've got P21, and now another negative regulator is RB, which stands for retinoblastoma protein. I know retino, I think of the eye. It has nothing to do with the eye, but maybe that'll help you. RB. I knew a guy named RB. Maybe he was a real drag. If you're watching RB, I don't mean it personally. But anyway, RB, what it does is so this is a transcription key, E2F. I mean, I don't know why it's called this, but anyway, this is like a key. When E2F binds to the DNA, it causes, okay, I'm turning on reproduction. And so what RB does is RB can serve like a magnet to this thing. Nope, I'm not letting you put the key on the DNA. We're not going to be transcripting any DNA yet. And so this RB, retinoblastoma protein, keeps the E2F from causing the replication of a particular, or the creation of a particular protein. It also monitors cell size. And this didn't make sense to me. It's the opposite of what I thought, where I thought they were going to it. But basically, RB, apparently, what it does is it says this cell's not big enough yet. Or you might say RB comes and says, you're not old enough to be reproducing yet. And so RB keeps E2F from triggering the reproduction. Until the cell gets a certain size, the RB keeps the reproduction from happening. But once the cell is a certain size, then it'll let it happen. And the way this is triggered is by phosphates. Phosphates basically pull the RB away from the E2F, which is allowed. I don't know, Fe. Think of Fe. E2F. Anyway, Fe. But anyway, so the phosphates then pull the RB, the retinoblastoma, away from the F, which allows the F magnet, releases the F magnet, and the cell cycle can continue. These are also, this is a tumor suppressing protein, and there's a question, why would it make sense for it to be a tumor suppressing protein? I guess because this didn't make any sense to me either, because I think of tumors as growing, so I'm not going to even hazard a guess as to why it is a tumor suppressing protein, because I don't know the answer yet. I just want to say that RB is a tumor suppressing protein. Okay, I think that's it. I think we made it. This has been 10.3, regulation of the cell cycle. 3 points were checkpoint Charlie. G1, is the DNA intact? At the end of G2, everything replicated here. And then at metaphase, are all the spindles attached? And so those are the three. There are positive regulators like the cyclins that hook up with the cyclin-dependent CD, cyclin-dependent kinases, CDK. So the cyclins hook up to the CDKs, and those are positive regulators. And then there are 3 negative regulators that were mentioned, Papa 53 and P21 and RB, retinoblastoma. Retinoblastoma. Okay, I should have stopped when I was ahead. This has been 10.3.